So it's my pleasure to talk about some recent connectivity work in my lab. So the focus of my lab is on the sense of direction or heading, which is essential for navigation. And we study this in flies by combining the virtual reality and physiology. So Vivek already told you some of this, but we tether the fly and put her in a virtual reality arena, and she is allowed to fly or walk on an air-floated ball. So when the fly is flying, we measure the difference between left and right wing beat amplitude, and we measure the rotating ball, the rotation of the ball when the fly is flying, when the fly is walking on a ball, to infer her intention of turning and change the visual scene to provide some kind of visual sensory feedback to close the loop from the motor command generation to the sensory feedback. So we call it a closed loop experiment. And I think Vivek already showed you some similar video, but I cannot be bored with this video, so I'll show you again. So Johannes actually expressed the genetically encoded calcium indicator, GCAM, in the entire population of compass neurons. So you saw these neurons in Vivek's talk, and I think Larry also showed this population. Anyway, so what you see here is that, so this is a view from the behind of the fly. So this is abdomen of the fly, this is the thorax, and this small structure is the fly holder, and the fly is walking on an air cushioned ball. And this is the visual scene projected on the LED arena, okay? And let's see what happens. So you are recording from the entire population of compass neuron here, and you can see that small patch of calcium activity follows the body orientation or head direction of the fly in the virtual reality arena, right? So in flies, we can actually see the compass needle, literal compass needle in the head. Both Vivek and Larry told you that these compass neurons in the fly forms a ring attractor. And this is a model developed about 30 years ago to explain the head direction system in mammals. And it was a big surprise that the structure was discovered, almost one-to-one correspondence of these neurons, these hypothesized neurons in the fly compass system. And that was a big surprise. And for today's talk, I'll just simplify it like this. So this individual small circle represents individual compass neuron in fly. And this blue arrow means that there is all-to-all inhibition, and there is an excitatory connection, recurrent connection between neighboring neurons. And this inhibitory connection among neurons will be important in the later talk. So I'll show you one experiment that I did to show that there is indeed inhibitory interaction among the compass neurons, okay? So you can see a small bump activity, calcium activity here in the population of compass neurons. And I'm going to stimulate here, which are basically quiet right now. And the goal is this experiment is to see if I stimulate or activate this part of the compass neuron, and the existing bump will disappear, okay? That's what we want to see. So we did that using optogenetics, and I'm stimulating here now, and you can see that new bump appears here, and the existing bump disappeared, right? And this new bump stays there even after I stop stimulating it. So let me show it again. The red rectangle means activation. Okay, the existing bump disappeared. So this is strong evidence that there's indeed all-to-all inhibitory dynamics going on among the compass neuron population. So this is the enough background, and I want to do a mental experiment. So close your eye and point to the door. Okay, you did this with Vivek already, right? So he stole my show already. Okay, that's it. You can do this very well. I have an upgraded version of mental simulation. So let's say you close your eye, stand up, and rotate like 10 times and point the door again. How many of you can reliably point to the door?
We did that experiment with
okay.
small ones, and half of the students were sick for several days. They were stumbling as if they were drunk. It's not a very recommended experiment.
Okay. Yeah, that's okay. Okay, I won't do that experiment. Interestingly, if you have the sound cue, you can still reliably do that. But if you muffle your ears, then you cannot do that very well, except for some very good people. Anyway, now you have your system actually accumulated a lot of error. But as soon as you see this kind of scene, you will instantly recover your sense of direction, right? So the point here is that I want to make here is that visual information is very strong influence on the sense of direction. But not all the features in the scene is critical for your navigation, right? The humans here are not important, okay? And you can easily imagine this situation. When you walk into a room, you can easily point the window, right? But it doesn't really matter whether the color is red or the location of the small objects like TV, right? So those information are not critical for navigation. And this can be seen in animal experiment as well. So this is an experiment done in rats. So this is a small arena. It's a top-down view. It's basically a small box, rectangular box. And you can see that there are some stripes on the wall, right? And this red circle represents the location where the food is buried under the ground. And the animal, the rat, is supposed to find this location and dig to get the food. And after about 2 or 3 trials of training, the rat is removed from the box and again rotated several times and get back into the arena. And the task is to find the food again. And of course, the rat will dig this place, right? But for the half of the trial, they actually dig this corner as well, which suggests that some visual pattern, like these stripes on the wall, does not provide much information about the spatial perception. And this can be observed not just in the behavior, but also in the physiology. So this represents the some kind of response pattern of a set of neurons called place cells. For those of you who are not familiar, place cells are responsive when an animal is on a certain location in the environment. Okay, in this case, this particular neuron becomes active when the animal is in this corner where the food is. But you can see that in other trials, the same cell is firing when the animal is in this corner, in this corner. And this is actually correlated with the behavior of the animal digging in this corner, okay? Meaning that, again, it clearly shows that there are some visual features critical for mammalian navigation, and it doesn't seem like this pattern on the wall, okay? And there are some followed experiments from this group showing that maybe the geometry of the environment is more important for mammalian navigation. Then the question is, for flies, in this case, what kind of visual features are used in the navigation system? And if we go one step further, how are they extracted from the visual input? And how are they integrated in the compass system? And that's where we get very useful information from anatomy. And this is a result from the reconstruction effort at Janelia using hemibrain data. And what you are seeing is a collection of compass neurons, which, okay, it's not moving. Okay. Which tile the ellipsoid body. And oh, that's strange. Yeah, it's almost manual play. Anyway, there is a yellow neuron. This is one of the compass neuron. And there is a red neuron like this. And this neuron is called ring neuron because of the shape of this neuron. And this ring neuron makes direct synaptic contact with this particular compass neuron, as shown in this electron microscopy data. Okay. And by the way, this ring neuron, this red ring neuron, single ring neuron, makes synapse with the entire population of compass neuron along the ellipsoid body perimeter. And this ring neuron is GABAergic and inhibits the postsynaptic compass neurons. And there are many ring neurons like this. Okay. There are about 12 different types of ring neurons, and total number of ring neurons is probably about 200. And overall, they effectively create an all-to-all connection from ring neurons to the compass neurons. And from previous studies, again, from Johannes, who discovered compass neurons, we know that many of these ring neurons respond to visual features at a certain location in the scene. So here, we are recording from small microglomuli. Each represents a single ring neuron. And we can plot the response pattern in this plot. So each rectangle represents individual ring neuron. And the red means the location of the visual field where the neuron is excitatorily responsive. And blue means that when the stimulation in blue region, the neural activity is inhibited. Okay? And you can see that these 6 neurons have a similar response pattern with different locations, by the way. Okay? So what visual features do they encode? A few years ago, David Bach at Janelia, and now he's in Vermont, finished collecting the electron microscope data of the entire fly brain, opening up the possibility to reconstruct single-cell resolution circuit structure. And we use this data to backtrace the visual pathway beginning from the compass neurons all the way to the eyes. So this is a result of a heroic effort of Dustin Garner, excellent undergraduate student. He reconstructed nearly 80% of this entire pathway. And it is also fantastic collaboration with the Matthias Wellness group and Alyosha at Janelia. So we reconstructed over 3,000 neurons using an AI-assisted EM reconstruction framework called Flywire from Princeton University. And it covers about 450 neurons that originate from the medulla and sends axons to the anterior optic tubercle. Because it begins from medulla to the tubercle, we call them Me2 neurons. And there are about 75 on each side, tubu neurons. Of course, from the name, you can infer that it begins from the anterior optic tubercle and sends axons to the structure called the bulb area, which is abbreviation is BU. And there is a ring neuron that has dendrites here in bulb and send axons to the ellipsoid body and make synapse with the compass neurons. With this data, we first categorized the pathway beginning from the Me2 neurons. I'm not getting into the details of today, but we used the connectivity information, morphological information, and anatomical locations of the processes and categorized these neurons into 4 groups. And these 4 groups of Me2 neurons innervated different areas of anterior optic tubercle here, shown from the top. So the question is, what kind of visual features are processed by each of them and downstream neurons? And for this talk, I'll just focus on Me2-1 and Me2-3. I mean, we were lazy to just assign the names 1, 2, 3, 4 to these groups. We didn't have cool name for each group. But anyway, we will focus on MIT1 and MIT3 because they show very different connectivity pattern to the downstream pathway. So this figure shows a single MIT1 neuron, and this is the top-down view of the single MIT1 neuron, and this is a side view in the medulla layer. And these gray circles represent columns in the medulla. And each column, you can consider it as a kind of pixel of the fly. And fly eye has about 800 pixels on each side. And you can already see that MIT1 neurons covers about 20 to 30 medulla columns, meaning that detailed visual feature is not really important for navigation. So the head direction system doesn't care about small details, but just some kind of blurred image of the visual scene. And this is the entire set of the MIT2 neurons on the left side. And if we pick 3 of them along the front to back axis, okay, then you can see that they are clearly arranged in the anterior optic tubercle from bottom to top. Okay, and if we assign colors to entire MIT2 neurons along this front to back axis, you can see that these colors, different colors, are actually aligned, clearly aligned along this bottom to top axis. So what does this mean? Because we have the entire visual pathway in our hand, we can do something interesting. First of all, tubu neurons are also, we found that, aligned along this bottom to top axis. Okay? But differently from the MIT2 neurons, these are just single neurons. Okay? They are about 10 to 20 neurons here. Okay? And there's no overlap, basically. And also, interestingly, each tubu neuron, in this case, the specific type of tubu neuron is tubu 8, and each tubu neuron is connected to a single ring neuron. This is one-to-one connectivity. So there is no, if there is a very minor interaction between neurons. So it is just one-to-one connection. Then using this data, we can backtrace the connection from each ring neuron to the MIT2 neurons. So here is an example. And we picked an ER neuron and backtraced the entire visual pathway that provides input to it. In this case, we picked a ring neuron that is downstream of MIT1. And this part, the type is ER4D. Okay, I have to explain a naming scheme a little bit. So these are ring neurons conventionally, but neuron name is ER, which means ellipsoid body ring neuron, I believe. So occasionally if I accidentally say ER neuron, that just means ring neuron. Anyway, as you see that these MIT2 neurons, let me play it again. So these MIT2 neurons are vertically aligned, right? Meaning that this particular ER4D neuron may be sensitive to a vertical stripe, right? And we can repeat this prediction process or reconstruction or backtracing process for the entire population of ER4D neurons. And this is the result, okay? And this is the summary cartoon. So you can see that the visual fields that ER4D neurons are responsive are all vertically elongated and aligned along the azimuth axis. So what about MIT2-3 neurons? It turned out that there are 3 subtypes of MIT2-3 neurons, and the details are not very important. But we know that, we know that this subtype has a very flat dendritic arbor, whereas some other types has some kind of protruding dendritic arbor to other layers of the medulla. But the key feature of this population that I want to talk about is actually MIT2-3C, third subtype of MIT2-3 neurons. And you can see that MIT2-3C dorsal has innervation in the dorsal part of the medulla. and mid-to-3C ventral has innervation on the ventral part of the medulla. So what does this mean? This shows the neurons backtraced from the ER2 neurons, which are downstream neurons of the mid-to-3 neurons. And you can see that the innervation is limited or constrained to the upper medulla or dorsal medulla. Whereas here is another example of mid-to-2 neurons backtraced to the mid-to-3 neurons, and you can see that the innervation of these mid-to-3 neurons are confined into the ventral medulla, right? And you can repeat this backtracing process across many ER2 neurons, and you can see that the actual innervation of the mid-to-3 neurons is limited, confined to a small circular region. And this is the summary plot, summary cartoon of this population. And you can see that smaller visual field that neurons are responsive actually tiling the visual field as a two-dimensional tiling. Okay? And we wanted to test these predictions in physiology. So coming back to this video, I told you that Johannes in Vivek's lab recorded from individual ring neurons and generated this kind of plot. But it turned out that this visual stimulus can only cover about 210-degree azimuth and 60-degree elevation, meaning that if you go back to here, the span of this single circle is about 60 degrees, whereas this one covers almost 180 degrees. So this experimental setup cannot really test our hypothesis. I'm using the same setup, by the way. So I had to modify the setup a little bit, which is very simple. So this figure shows that. So this small elongated visual location of this particular neuron may be actually this long, but this cannot be tested in this setup. So we modified the experimental setup a little bit, which was simple. So Jennifer simply tilted the head to expose more part of the eye. And we used some projector-based screen to stimulate a larger area of the eye. And this is the stimulation profile viewed from the fly. And this is the projected screen stimulation, projected stimulation on the screen. So this plot shows the results. So for ER4D neurons, which are downstream of the mid-to-1 population, indeed shows highly elongated, vertically elongated visual field that, and the neurons are responsive to this field, whereas the ER2 neurons that are downstream of mid-to-3 neurons has smaller receptive field, a smaller visual field that they are responsive to. And we repeated this across many neurons, and you can see that all the visual field that ER4D neurons are responsive are vertically elongated, just like predicted from the electron microscopy Connectome data. And ER2 neurons are responsive to a smaller visual field. Okay, so this is all expected from the connectivity data and confirmed by physiology. But because we have more data, we didn't stop here and actually reconstructed further upstream of the mid-2 neurons and identified the neurons that provide major input to the mid-2 neurons. And there are PM2 neurons, MTI neurons, and mid-to-1 neuron actually has some recurrent connection among themselves. For mid-to-3, the input is MI15, MTI, R7, and so on. So the problem here is that all of these neurons have not been characterized except R7, which is a photoreceptor neuron. And it turned out that, R7 neurons are sensitive to the UV and it is inhibitory in flies. So it strongly suggests that these ER2 neurons may be inhibited if there is a UV signal, a UV stimuli, right? So we tested this in the same way. So this projector was originally only projecting blue light, but now we are using UV and blue and amber stimuli. And as predicted, when you characterize the response pattern of these neurons, you can see that ER2 neurons are totally inhibited by UV stimulus. Whereas, to our surprise, ER4D neurons are agnostic to the wavelengths. It is responsive to UV, blue, and amber light. So in summary, I've talked about two anatomically distinct visual pathways. And the ER4D pathway process the vertical stripes of all colors, whereas the ER2 pathway process two-dimensional arrangement of a scene in blue and amber. So we can change the plot like this. So we have UV inhibitory property here. But this is not the entire story. I just showed you two examples out of 12 potential visual features. So Me2 have about 10 subtypes in total. Tubu neurons have about 10 subtypes. And ER neurons or ring neurons have at least 12 subtypes. And I just showed you two of them. And another important pathway, for example, from Me2 is Me2 pathway is that they encode the skylight polarization, which is critical for insect navigation. And we also know from the connectivity data, this skylight polarization pathway and this Me2-3 pathway have strong inhibitory interaction with stronger inhibition from skylight polarization pathway to this Me2-3 pathway, meaning that in the navigation, during the navigation, skylight polarization may provide stronger cue for head direction. So these are some other predicted field of the predicted visual field that each ER4M neuron, the downstream of Me2 neurons, may responsive. And this is the response pattern, potential predicted response pattern of these ring neurons, which are downstream of Me2-4. Unfortunately, we don't know what Me2-4 pathway, we don't know the visual features that Me2-4 pathway encode. And I think this is kind of a project that I think Srini can help us to predict some potential visual features. Okay, so I told you that there are many ring neurons connecting to the compass neurons. And overall, effectively, it creates all-to-all connections from ring neurons to compass neurons. And if you see the synapse count, by the way, please ignore this EL type because it's not relevant for today's talk. So if you see the synapse count from ER4M neurons, which are sensitive to skylight polarization, to the EPG neurons, which are the official name of compass neurons, you can see that synapse count has some kind of variation, right? Meaning that you can actually predict the preferred skylight polarization pattern that each compass neuron may prefer, right? But the story is very different for ER4D and ER2 neurons. So these ER4D neurons that are sensitive to vertical stripe, it has almost a uniform connection to the entire population of EPG neurons or compass neurons. ER2 neurons has the same kind of property, meaning that these two-dimensional organization encoding population has the uniform connection to the compass neurons, which poses a problem because compass neurons are sensitive to, encodes the body orientation, right? When you change the body orientation, visual scene changes, but the inputs to the population of compass neuron is potentially the same if the uniform synapse count actually represents uniform synaptic weights within the same population. Of course, it's not. So the answer to this question, whether the uniform synapse count represents uniform synaptic weights, the answer is no, they are not. And I'm going to show you clear physiological the evidence of this thing. And of course, this is not surprising thing. So to set the stage, let's see this clip from Ready Player One by Steven Spielberg. The protagonist, this boy of the movie, is in a small van, and he will be instantly transported to an amazing virtual world. I mean, this is not sci-fi anymore, right? So he is now in a new world, but it won't take him more than a few looking around like this to get his stable sense of direction, right? This illustrates two important things. Your sense of direction is very flexible, first of all. And once adjusted to a new environment, it quickly becomes stable. So it is easy to see why this flexibility and at the same time stability of the head direction system is a kind of shared need across the animal kingdom. And we are interested in how the brain flexibly generates stable heading representation across different scenes. And as before, we recorded the activity of the entire population of compass neurons. In this case, you see a fly from the bottom, and this horizontal feature is the fly holder. You can see the shade of the wing, which is not clearly visible in this video, but we measure the difference between right and left wing with amplitude, and infer the fly's intention of turning and change the visual scene surrounding the fly. And this scene is from this natural photo, and it is monochromatized and downsampled. Okay? And you can see that when the fly turns within this scene, the bump actually follows pretty well, right? So it encodes the fly's heading inside the scene well. And notice that there is a very clear relationship, angular relationship between the scene orientation and the location of the bump. So we call it an offset. And this offset is consistent within a fly, but can vary across flies. So how can I say? For this particular fly, for this particular scene orientation, the bump is around here, as you see here, right? But if you repeat the same experiment in other flies, the bump location will be at 1 o'clock here or 9 o'clock, right? So it varies across flies, but within the fly, it is consistent. So it's like, okay, so everyone here experienced this room probably for the first time on Monday. You experience basically the same experience, but the representation of the space is unique to all of you. They are different across individual. And that doesn't really matter, right? As long as it encodes the heading direction consistently, the rest of the brain will be fine with that. So the question is, why do they vary across flies? And one potential hypothesis is that this mapping from the visual scene to the bump location is experience dependent. So we tested it. Again, for this testing, I want to point out that there is all-to-all inhibitory interaction among compass neurons. So if you stimulate one part, the bump will be shifted to here, and the original bump will disappear. Using this property, we tested this experience-dependent plasticity in this population. So here, you see that the fly is flying in this scene, and the bump is following the scene orientation. And for this particular scene orientation, bump is at around maybe 530. Now I will enforce the pairing between the bump location and the scene orientation. Okay? It's not a closed loop experiment anymore. So for each scene orientation, I enforce the bump location using optogenetics. So for this particular scene orientation, which is the same as this one, I enforce the bump location changed from here to 12 o'clock. Okay? And I repeated this experiment like 5 minutes. It's not long, just 5 minutes. And you can see that in this probe trial, for the same scene orientation, the bump is at the top right now. Okay? So this is pretty strong evidence that the encoding of the head direction is experience dependent. And the idea behind here is also pretty simple, right? So if two phenomena happens together, and then the connection between these two phenomena becomes strengthened, which is basically Hebbian plasticity. So we developed the model, and to explain the model, I need to go back to Johannes' experiment. So the fly is now in a complete darkness, which kind of represents the situation when the visual input does not provide any meaningful directional cue. The fly is rotating the ball, and the bump is actually following it, meaning that there is an additional information that is generated from motion, the self-generated motion, that updates the location of the bump. And this process is critical for the modeling. So here is a model. So I told you that the ring neuron population has connection to the entire population of compass neurons. And for the sake of simplicity, I aligned the compass neurons along this vertical axis, and I aligned the visual neurons on the x-axis. In this case, visual neurons should be visual ring neurons, and they are inhibitory neurons, but for the sake of simplicity, again, I just assume that these are excitatory neurons. And vertical lines represents the axonal process of the visual input neurons. Horizontal lines represents the dendritic process of compass neurons. Red means high activity, gray means no activity, and the cross means the synapses between visual input neuron and compass neuron. And in this case, in this particular case, a few visual ring neurons, this one and these three, are active for this scene. Okay? And because of the ring attractor dynamics, small number of compass neurons are lumped together here, and they are active. Okay? If we begin from some random activity, they will collapse onto a single bump of activity like this. Then the connections between these active neurons are strengthened. And the size of dots here represents the strength of the synaptic connection. And I hope this makes sense, okay? So if two sets of neurons, this one and this one, two neurons are co-active, then the weight between those two neurons becomes stronger. And that happens in other connections between active neurons, okay? Now, this fly flies around, okay? And at some point, it may be looking in the opposite direction. Then a new set of visual neurons will be active, which are 180-degree shifted. At the same time, because the fly turned 180 degrees, the bump of the compass neuron would have shifted 180 degrees, right? So the connection between these visual neurons and these, these visual neurons and these compass neurons will be strengthened. Does it make sense? And the fly keeps flying around, and this process keeps strengthening the relevant connections. Then eventually, the information from the scene will be fully engraved into the network. That is, the connection between the specific orientation of the visual scene and corresponding compass neurons has been established. And this is a simulation. So at the bottom, I show the activity of the ring neurons, which is, I'll probably end earlier. Yeah, so here, the activity represents the compass neuron activity with some added noise, and y-axis here represents the activity of the compass neurons, and it began with the random activity. And this matrix of dots represents the synaptic weights from the ring neurons to the compass neurons. Initially, we randomized the matrix, and you see that the synapses between active ring neurons and active compass neurons are weakened over time because, in this case, I correctly assumed that ring neurons are inhibitory. So if the connection becomes weaker, it actually means the pairing becomes stronger. And other synapses will be strengthened through potentially some homeostatic process. And we ran this simulation with the real behavioral data we collected from flying flies. And you can see that a band of weakened synapse appears over time. So I should say that the basic idea of this model has been proposed in many different contexts, including head direction cells, grid cells, and the actually insect central complex as well. And also the mathematical formulation is basically identical to a system called one-dimensional self-organizing map. And what is really cool about the fly central complex is that we can test some crazy predictions of this model in vivo. So one of the crazy prediction is this. So if it is really experience dependent, then we can mess with the experience to mess up the mapping. So in this case, you can see that the bump rotates clockwise when the scene rotates clockwise, right? Now you can enforce the mapping on the opposite direction so that when the scene rotates clockwise, the bump is mapped onto the counterclockwise. Then in the probe trial, you can see that when the scene is rotating clockwise, the bump is rotating counterclockwise, meaning that when the fly turns left, they feel they are turning right. And this is quite strong evidence that the mapping depends on the experience. So the answer to this question is, of course, no. And I'm sorry for many graph theorists here. So the actual biological network is more complex than just synapse count. Okay, so in summary, the compass neurons receive visual input from 4 major pathways, and the connection, I think the battery is out. So the connection from the ring neurons to the compass neurons is flexible. Okay. I didn't talk too much about the ring attractor dynamics about the compass neurons, but if you are interested in, I can talk about it after the talk. So yesterday, Gwyneth showed this slide that I liked a lot. So she said that the connectomes are democratizing, which I wholeheartedly agree because I have never thought about myself getting into the anatomy field, but I accidentally getting into here and presenting something in Connectomics conference, which is totally unexpected. And the second one is that connectomes let us think on a brain-wide scale, which is also true. One of the big questions that I'm interested in is to connect the perceptual neuroscience to the cognitive neuroscience. In other words, I'm interested in how the sensory information is transformed into a cognitive function. And this connectome data allowed me to explore some potential mechanisms using the fly. And the third one is that connectome enables a different approach to experiments. And you saw really small scale difference, which is just tilting the fly head to expose the eye. And that allowed us to test the experiment, test the hypothesis. And fourth one, connectome reveal new functional motifs. I would say rather than new, I'd say, many functional motifs because computational neuroscientists are smart enough to figure out most of the small motifs already. And you saw a lot of examples in this conference. And finally, Connectome bridge theory and experiments feeding conceptual level insight. And you saw a really good example in Larry's talk, right? So that combines the theory of the navigation to the structure of the fly brain. So with that, I thank all the lab members over there. And it is a wonderful, the first part of the talk was the wonderful work of Dustin. And physiology data was collected by Jennifer. And it was a great collaboration with people in Germany, in Janelia, and Larry. That's it, and I'll take questions.